Hi, and welcome to Lambda U. My name is Andy and I'm a Solutions and Support Scientist at LICOR. Our previous course was all about the primary antibody incubation and washing steps of the Western blot assay. Now we've covered those steps, the next part of the process is the secondary antibody incubation. In this course, you will learn all about secondary antibodies and how they are made, how to choose the best ones for your needs, the protocol for this step, whether you are using one or multiple antibodies, and some tips and best practices for how to perfect the technique in your lab and achieve the best results every single time. Let's start with what secondary antibodies are, how they are made, and how they fit into the Western blot assay. Secondary antibodies are always polyclonal, and they are produced in the same way as polyclonal primary antibodies, except the host species is instead immunized with antibodies from a different species. The secondary antibody, which will recognize and bind to the antibody from the other species, is then isolated from the blood serum of the host. For example, rabbit anti-mouse antibodies are produced by injecting mouse antibodies into a rabbit, and then isolating the anti-mouse antibodies produced by the rabbit's immune system from its blood serum. Goat, donkey, and rabbit are commonly used host species for producing secondary antibodies, but others may be used as well. You can learn about secondary antibodies in much more detail in the Lambda U learning path, The Importance of Antibodies, but for now, let's look at how they are used in the Western blot workflow. Secondary antibodies for Western blotting are labelled in some way, and this allows them to be detected and usually also quantified using an appropriate detector. When they are introduced to the blot, they recognise and bind to the primary antibody on the membrane, and this in turn allows the target protein to be detected. They bind to a region on the primary antibody that does not interfere with the primary antibody's binding to the antigen. Because the secondary antibody is made in a species which is different to that of both the primary antibody and the target protein, non-specific binding is minimized. Secondary antibodies are often also affinity purified to ensure high affinity and specificity for the primary antibody. As discussed in the blocking course, using a secondary antibody also serves to amplify the signal when compared to using a labelled primary antibody alone. This is because multiple secondary antibody molecules will bind to a single primary antibody molecule, and this will cause approximately a 3 to 5-fold increase in the number of labels indirectly bound to the target, and consequently a significant amplification in the signal. Secondary antibodies can be conjugated to a number of different labels for detection, but the most common are enzymes or fluorophores, also known as dyes. In the next lesson, we will discuss the advantages of using dyes for detection, the different types of dyes that you can choose from, and some advice on choosing your secondary antibodies.